The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseWare continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseWare at ocw.mit.edu.
Two things I want to do today, just briefly go through Chapter 6, which is about brain specializations. It was meant to be sort of an interlude. It was a little bit of a break before we get into some of the forebrain connections. But these all involve specializations because of the evolution of head receptors and the resulting expansions of the brain that have occurred. And we start with electroreception in some fish that's led to this enormous expansion of the cerebellum in the marmorids. And my first question here was, what cranial nerves carry information from electroreceptors in fish that have electroreception? And also, why is it needed? Why don't they use vision? Yeah. They're cranial nerves, that's right. But what cranial nerves are they? Sorry? There's actually up to 6 of them. And what are they called? The lateral line nerves. Because fish have these receptors along a line along the side of their body. They also have mechanical receptors there. There's a mechanical lateral line row of receptors also. But we're concerned here with the electroreceptors. Okay, so what are they doing? Why is it so important? Why aren't they just using vision? Yeah, go ahead. Yeah, because they can't see. They're living in muddy waters. They thrive in those waters, but they've got to be able to find things. They've got to be able to find prey if they're feeding on them. So what are they detecting? There's 2 kinds of electroreception. Let's just deal with one of them. The fish that generate an electric field around their body. It's not a big electric field. There are a few of them that can generate such a big jolt of electricity that they can stun prey. But we're not talking about those. We're just talking about them that use it as a sense. They generate an electric field, but they also detect the electric field all around the side of their body. So any disturbance in that field, they can detect. So all that computation involved in this specialized brain here, the cerebellum, this is the area in one of the marmorids, are involved in computing where the animal is in order to get a particular disturbance in the electric field. It's not an easy task. And it's a huge computational problem, and there are computational neuroscientists still trying to figure that out. All right. No placental mammals have electroreceptor abilities, but one non-placental mammal does have such an ability. Do you know which one? It is in the chapter. The duck-billed platypus, yes. Okay. How are this animal's electroreceptor inputs different from electrosensory fish? They use a different cranial nerve, and somebody said it. So, anybody else know? I think you said it before. It's trigeminal nerve, that's right. But not in those fish, just in the duck-billed platypus. It's in their bill. So how do they feed? They stick that bill down in the muck and in the water. And they're detecting electric fields. Many animals do emit some kind of electric fields. I don't know the details for the duck-billed platypus. I know a little more about the fish. All right. Another sense that is not highly developed in mammals is infrared detection. In what animals is this particularly important? And what cranial nerve is involved? Anybody else? Somebody that's not talked today yet? The pit vipers, yes, snakes. But only particular snakes. Snakes, like the rattlesnake. So here, in this little picture, I think this is the same one I used in the book. I've added to the picture I found in a drawing in the Hodos and Carton book. But this is the pit. It literally is a pit. It's recessed. What would be the value of that? Why put the infrared detectors at the bottom of the little pit in their face? Yeah, because you just imagine, I have a cup here, and the receptors are back here. Now it's got directionality. Now it's not responding to things over there. It's only responding to things in this direction. So they have to move their head, of course. It doesn't give them directional selectivity if they keep their head still, except they know it's right in front of them. All right, so, and you can see it comes in through a branch of the trigeminal nerve to a particular group of secondary sensory cells, and then through a circuit involving the trigeminal neurons, it goes from there to the optic tectum. Remember, we call it optic tectum, but it actually gets many different sensory modalities, distance receptors. It gets auditory, somatosensory, and visual, and now we see it gets information in these snakes from their infrared detection. So they can use it to orient towards their prey.
Does every type of snake have?
No, only the pit vipers have that. And it would be interesting to look to see what other kinds of infrared detection you have in animals. I'm sure other animals have evolved that. It's too useful not to evolve only in one small group like that. But okay, bats are not the only mammal with echolocation ability, another special sense. So you'd expect specializations in the brain of the bat. But what other mammals have echoreception ability? And the dolphin. Okay, and what cranial nerve do you think has expanded? I've actually studied that with Eric Monti, who's a marine mammologist. We did, using MRIs, we were able to reconstruct the brain of the Atlantic white-sided dolphin, and it was confusing at the beginning because I had never seen, I was helping him identify everything, and I thought it must be the trigeminal nerve. Any mammal I've ever seen, the trigeminal nerve is the biggest, but not in that animal. It was the eighth nerve, okay, the auditory nerve. And similarly, if you look at the surface of the midbrain in the dolphin, look at this enormous inferior colliculus. The superior colliculus is relatively smaller, and it's the same in the bat, at least very similar. The dolphin, of course, has an enormous end brain too, and so I've taken a picture there where the end brain's been removed in the dolphin, but not the bat. Yeah. Sorry? As humans that use some echolocation, it does become specialized, and there are indications that blind people that are more likely to be able to use such an ability do have changes in their cortex. And we're going to talk a little bit about that towards the end of the class. It's a very good question. Just notice here the other two animals in this picture. It shows animals where the superior colliculus, the more anterior surface of the midbrain, is enlarged. And they're very visual animals. Here's a goat, a wild goat, an ibex, and tarsier, a prosimian primate that can literally locate using that structure. It's got this innate ability to orient and reach out with his hand and grab insects literally right out of the air. All right. What is a brain manifestation of the specialization of primates for vision? That includes us. Most of the primates have they're very specialized for vision. That's why monkeys are used so much in studies of vision, because of similarity to humans. You know, rats are used too, and cats used to be the most popular animal, but no longer. It's because of this specialization. So you see, what has expanded so much? Isn't that tectum? It's not like in the ibex and the tarsi here, but it's a part of the brain more involved in visual learning. All right? And this shows an owl monkey. There's the striate cortex, what we call primary visual cortex. But every area that you see outlined with the black line, and many of them are also in different colors, are separate representations of the visual field, or separate in some way in the electrophysiological recording studies. They all have at least a partial representation, separate from the others, of the visual field. So you can see how the visual areas have expanded into the posterior parietal region and the temporal region, from the middle temporal area here all the way down into the inferior temporal lobe. Okay. What's the specialization of small rodents? They're using their whiskers. What's happened in the brain of these animals? They have a specialized sense too. But it's not like any of the others we've just looked at. And we don't have it. The barrel fields, each barrel, named because of the way it looks in the histology, represents one whisker. And these are sections of the cortex that have been, they try to flatten the cortex and cut it, or at least they cut it tangential to the surface. And here they've taken different levels, and they go down through this area of the parietal lobe, and they see these barrels. We'd have to blow that up to see how these really look like barrels. They're crowded neurons, packed neurons around the edges, and very few in the middle. And if you look in the middle of the barrels, all the cells would be in the dark areas here. This is stained for axons. You see, the axons from the thalamus come up, representing each whisker, coming up in the middle of those barrels and terminating. So it's a very specialized organization. It's been very useful for electrophysiological studies. Other somatosensory specializations exist too, like the hand in apes, and raccoons, and certainly humans, because we have such high acuity and motor control. But also expanded cerebellum for coordinating those movements. And I mentioned here the prehensile tail in the spider monkey, and you get expanded representation of that tail in sensory and motor neocortex. And this just shows the raccoon. This is a picture from the book, and it's comparing the quadrimundi and the raccoon. They differ tremendously in their use of their forepaw. The raccoon literally has a sensitive hand, just like we do. And if you look in the cortex, this whole area outlined in red there, here's an enlargement of part of it. There's a separate little gyrus for each digit. They have very fine sensory ability and corresponding motor control of that hand. Whereas if you look at the quadrimundi, who also uses his forepaws, as many animals do in feeding, the corresponding area is much smaller. And he doesn't have a separate gyrus for each digit.
Does the raccoon
He does have enhanced sensory abilities. Yeah. And what may be a brain specialization of the human ability for complex social interactions? And this would apply to many other primates that live in groups with complex social behavior. What do we think? There's an increasing amount of work on this now, especially the people studying the social brain here in the building. Yes? In the neocortex, especially prefrontal areas, which is frontal lobe association areas. We'll be defining all that when we later. These are the areas that they keep localizing higher functions to, and we'll talk about their evolution. And I just want to point out, I want to ask you if you remember what the specialization that I illustrate in the book, in the echidna, which is the spiny anteater of Australia. He's got an enormous prefrontal cortex. Why would he have that? You think it has to do with it? This is the pink area here. It's relatively larger even than in humans, just in relative terms. In the colors here are the motor area, sensory area, visual area in green, and the darker green is the auditory area. And these are, we would call them association areas. But look at that prefrontal area. So it has, I assume it must have to do with his specialized feeding. He's an anteater, and ants are very small. How does he maximize his intake? You know, I think there's tremendous demand on his working memory. In a single glance, he has to remember where dozens of ants are, where they're disappearing, and he's got to be able to go after them. But that has never been analyzed. Okay? One way to get it to be analyzed is to put it in a book and raise the issue. So people will look at it. This is a question for you. What other specializations might have noticeable brain manifestations? I want you to think of something that I didn't mention in the chapter. What other specializations in animals that you know about must have some special brain representation? Sorry? Anybody? Anybody else? Okay. That's never been investigated? Do they have a specialized vestibular system? What would you expect in that case? Anybody else? Specializations. I asked, yeah, go ahead, Rachel.
I know that taxi drivers have a really well-developed, is it amygdala?
Parietal neocortex, parietal associations, the area where we retain long-term memories, the map of the environment. That's right. There is some evidence for that. It's a correlation, and it's difficult to prove cause and effect, but it's a very interesting correlation. These are London taxi drivers. We know that the cortex is affected by learning in pretty interesting ways, some of which are still being discovered. I asked my wife this question this morning, and she came up with specializations in the nose of some animals. What was the one you thought of? The star-nosed mole. John Koss actually turns out to be one of the guys who's wrote an endorsement of my book, so I like John Koss, and he has studied this star-nosed mole and published representation of that. It looks like almost like a hand, multiple fingers. It's on their nose. And they use it when they go through the ground. Okay? And they use it for feeding. Yes? That's another thing that we'll be dealing with a little bit, but you're right. There are changes that occur in blind people, and that's been studied in animals. in very interesting ways. And I would just add the one other animal with a specialized nose, the elephant. You know, he's got incredible motor and sensory ability in that nose, in that trunk. All right. This is the bigger class. We won't quite get through it today, but we'll do our best. I want to give you an overview of forebrain structures and introduce the neocortex. I want to start with this question: what can an animal do without a forebrain? And this has actually been done. It's a pretty gross lesion. You know, they either disconnect the forebrain from the midbrain with a cut. In some cases, they leave some of the connections. So it's been done. The initial studies were done in the cat, but they were done on rats and pigeons. It's all in early literature. And let me just go through some of that. You call that a decerebration. Basically remove the cerebral hemispheres or completely disconnect them from other parts of the brain. So this is our earlier diagram, and what I'm showing here is the division: spinal cord, hindbrain, midbrain, and everything from here on is the forebrain. But the cerebral hemispheres are everything in front of this line here. This is thalamus and hypothalamus, they're tween brain. So they're disconnecting all of these structures. And of course, in the mammals that they're doing it, this area has expanded tremendously. The neocortex has grown. Okay, so they get rid of all of it. Why do you think they got huge differences between the results in the cat and in the rat? They both are mammals. They both have a neocortex. They both have a corpus striatum. You know, and in both animals, they disconnected those areas from the lower brainstem, or from the brainstem, period, from the entire brainstem. They got big differences. What is the main thing I claim in the book is the difference? Because the early interpretations were not this. And there's been all kinds of arguments about it. Yes?
The number of connections severed.
Yeah, the quantity, the number of connections severed. Very good. Why would you be disconnecting so many more connections if you're dealing with a cat than in a rat? They have the same kinds of connections in qualitative terms. But think of the size of the cat hemispheres. He's got the highly folded cortex. He's got a relatively large neocortex, whereas the rat's a smooth-brained mammal. Much smaller in relative terms, a much smaller cortex. Okay.
Does this mean that it would cause more damage in the rat?
Well, that's what we're going to consider. Why? Why is that? I claim it's quantitative. Let's go through this. But there's another question here. Actually, both rats and cats, the only way you can keep them alive after this kind of lesion is to force feed them. Why was that the case? They couldn't keep themselves alive. Does that mean they weren't hungry? Well, in a sense, that's true. They never showed signs of hunger. But if you put food in their mouth, they ate. So it's not true that they wouldn't eat. They just didn't show the signs of hunger motivation. Okay? Now that the extensive study of decerebration in the cat was done by these guys, Bard and Mock, published in 1958. They called it the animal a purely reflex animal. All he had was the reflexes, they said. He couldn't do any act that requires a series of reflexes. That's the way they put it. They were still very affected by the SR model of behavior, and they thought they just couldn't put the reflexes together. We would say it more broadly now in terms of sensory-motor responses. So let's look at what the animal can actually do. And can it learn anything? First of all, the cats were anosmic and blind, because those are two forebrain senses. The cranial nerves for olfaction and vision come into the forebrain. They're disconnecting the forebrain, so of course, vision and olfaction can affect their behavior. They didn't eat spontaneously, as we said, but they also didn't groom themselves. They showed no spontaneous sexual or other social behavior. They could, though, elicit sexual reflexes by stimulating the genitals. They could get the lordosis response from females. They could get penile erection, but it required stimulation. They didn't show it in normal behavior. Okay, let's look at what they could do. They could stand, they could sit, they could right themselves if they tipped over, they could walk. They did show some abnormalities. They even showed rage, but only if you pinched them, especially their tail. They didn't bite or strike out normally. They showed some autonomic regulation. They showed piloerection. They would fluff up their hair in response to cold. They had some thermal regulation. It wasn't as good as normal. Their temperature would fluctuate more with changing temperatures of the environment, but they still had some thermal regulation. So commonly, if they wanted to have an easier time maintaining these animals and not have to give them injections of electrolytes and things, they left the hypothalamus attached to the pituitary. Even though it was disconnected from the midbrain, they left that hypothalamic island attached to the pituitary. And then they had much better regulation of their internal environment. They couldn't learn much. They had conditioned eye blink, conditioned respiratory changes, but they didn't maintain it very long. So what was it? I just said this. Describe the behavior that shows how a cat without a forebrain shows no hunger and yet will eat when you put food in its mouth. So what's he doing? He's responding to the stimulation. He will open his mouth, touch his lip. He will close his mouth on the food. What does that if he's not hungry? Sorry? No, I wouldn't say it's procedural memory. I would say it's innate reflexes. Okay? Exactly. Like the baby suckling. Okay? Except, babies, the suckling will vary with how hungry they are. Here, it didn't seem to. But that's the question. Maybe it did vary a little bit with how hungry they were. But the point is they didn't show any motivation. Yeah? That's a big issue in how the dopamine pathways, the role they play in feeding. It's the representation that we call representation of the motivational state, the drive state, is no longer, it doesn't mean they don't have it if they left the forebrain there, but just disconnected it. But it means it can't influence the midbrain and hindbrain and spinal cord anymore. All right. So then, this just summarizes if you do the same thing to a rat, he can do more. He's faster to recover his writing reflexes and locomotion. I said the cat had writing reflexes and locomotion, but in fact he lost it for a while. It took him a long time to recover. The rats recover faster. They did show some eating and drinking responses, but again, and more than the cat did. It was a little more than the normal, just simple reflexes, but they didn't seek food, so it was still fatal if they didn't force feed them. And now the rats would groom themselves. The cats never did. So, against that barred and mock conclusion, they did show a series of, you could call it a series of reflexes, if that's the language you want to use. We would describe it as a fixed action pattern, an inherited pattern of behavior. They also showed some defensive behavior that went beyond what the cat did. They would vocalize, they would try to escape, they would claw and bite. They even showed some auditory localization in space. Remember, the audition is the 8th cranial nerve, and it comes into the hindbrain, and it can reach the midbrain and cause orienting movements. Why did the rat do that, not the cat? So some people said the cat's forebrain has taken over more of the function than the rat forebrain. That as the upper parts of the brain involved, especially the cortex, it takes over lower functions. That was the way people thought about it. And I'm asking, is that the best way to describe the results? And I say, no. So some people said, well, you take the cerebral hemispheres away, they lose all their learned behavior. Let's look at that by looking now at another animal where decerebration has occurred, and that was in the pigeon. These were earlier than the other studies, earlier than the cat and rat studies. Okay, they were done by Dutch people, and it was all written in German, Visser and Rademacher. And 1935 and 37 they published this. They showed, after the decerebration, they showed their basic repertoire of unlearned reactions. They would fly if you threw them into the air, but they wouldn't start flying spontaneously. You had to throw them into the air. And while they were flying, they would avoid vertical sticks. Of course, this was all done indoors. They would land on horizontal sticks, but then they found out they would land even on the back of a dog or a cat. So what's happened? They forgot. Dogs and cats can be dangerous to pigeons. There was a lot of indication that more complicated behavior patterns that depended to some degree on learning during development were lost in these animals. So my hypothesis is that the forebrain is very important in linking together by learning the species typical action patterns, fixed action patterns that are built in, and therefore genetically determined. But we still learn to link those things together. We inherit the ability to do this and to do this, but that doesn't mean we can, we learn how to grasp a corner. That doesn't mean we learn how to play the piano. It's not innate. Just the movements are, but we link them in different ways by learning. And that's mostly a function of the corpus striatum. But first I want you to understand Nauta's terms. He uses these terms: stability of the internal environment, stability in space, and stability in time. And they involve different parts of the brain. So without a forebrain, Nauta said they still had pretty good stability in space, meaning they could balance, they could right themselves, they could move around. They had quite a bit of stability of the internal environment, but that was much better if the hypothalamus was left intact. But their stability in time was really messed up because their behavior is just their actions seemed to depend on the current inputs. They had little or no motivation-initiated behavior, little or no long-term memory. And of course, when you're dealing with mammals, they lose a lot of sensory and motor acuity. They don't have fine movements. They can't respond to detail the way they can with the cortex. If you spare the corpus striatum, they can do a lot more. I'm not going to spend a lot of time on that. You can read this. It's all in the chapter. They've also varied, instead of taking out the whole forebrain, they just do decortications in various animals. And the basic conclusion of those studies is that the more the cortex you take out, the more deficient the animal is in its sensory motor control. Again, a lot depends on quantity. As much as people tried to explain the behavior, the effects of these lesions in terms of specific cortical areas removed, and there was some correlation, but it seemed that the biggest factor was just the amount of cortex they took out. So that leads us to explaining this term invented by von Monaco a long time ago when he discussed corticospinal diastasis. Some very interesting analyses. What is meant by that term? What does it mean? Diastasis. You know what a schism is? Cut something into two parts. Diastasis, separation of two things. So I want to know, why is understanding of this phenomenon so important in interpreting these species differences in brain lesion effects? And in fact, in interpreting recovery of function in the human beings with brain damage. And that's why I think it's so important to understand it. In simple terms, it means deafferentation depression. If you deafferent a structure, it means you remove the inputs, or at least a lot of them. It's always a partial deafferentation. But with greater deafferentation, the greater the effect on those neurons. So, for example, take the phenomenon of spinal shock. Do you know that term? Have you ever heard that? Has anybody ever heard of spinal shock before? What is it? But here, you're thinking of deafferentation as removal of sensory input. And you're right. That will cause deafferentation of the dorsal horn neurons that are getting those inputs. Now let's talk about spinal shock. It's a lesion of the spinal cord. If I get a spinal cord transection, say at the bottom of the cervical region, okay? I don't just lose control of my legs and much of the control of my arms. No, I lose much more. I lose even spinal reflexes. I don't even show withdrawal reflexes or stretch reflexes. The reflexes go. But wait a minute, they're spinal reflexes. That's called spinal shock. It takes a long time for spinal functions, simple spinal reflexes that don't involve pathways outside the spinal cord to recover. So here's my diagram of it. Here I've taken two species. I'm showing something like a cortex here, and an animal with a lot of it, and an animal with less of it. And here's a little diagram of the spinal cord, and I'm showing a reflex pathway. I just show neurons coming in and other neurons going out, and they're connected in some way in the cord. Okay, and then you do this. You eliminate all those corticospinal connections. This is really a simple way to depict what von Monaco described when he first described diastasis. And he talked about corticospinal diastasis. I just put into diagrams what he was saying. I'm showing excitatory connections. We'll ignore the inhibitory ones for now. A lot more excitatory connections removed from the spinal cord in the animal with the larger cortex. Okay? So what happens? All these fibers are degenerated. These neurons are less deafferented in the smaller brained animal than these. That means the diastasis effect, the degree of deafferentation, is greater in the larger brained animal. So his spinal cord reflexes, the spinal reflexes, will take much longer to recover. If I transect the spinal cord of a frog, in not many days his spinal reflexes will recover. If I do it in a dog, it might take weeks. And if I do it in humans, it might take months. Just to get the spinal reflexes back. Why? Because the neuron can fire action potentials only if there is a depolarization of the membrane at the axon helix that reaches a critical level. You know that from basic physiology. Okay? If you remove a lot of its excitatory inputs, then there will be more inhibitory inputs from spinal interneurons that are left, and you just can't bring the cell to threshold anymore. Okay, so then what happens after such lesions? These two things happen. There's collateral sprouting in the cord. Collateral sprouting by remaining inputs. There's also what we call denervation supersensitivity. That means the neuron adjusts by increasing the number of receptors that respond to the neurotransmitters of the axons that are now missing. So it will respond more strongly to other inputs that use the excitatory neurotransmitter. Okay, so if this animal will show some sprouting here. This animal, because he has more deafferentation, will show more sprouting. And there will be more receptors on these neurons. And it will be more on the animal with the larger cortex. You'll get more supersensitivity. Those are the two factors. And just over the time course of collateral sprouting that we know about and that has been studied, you get quite a bit of recovery. Okay? From this deafferentation. So basically these quantitative effects have to be considered. Whenever I get a brain lesion, hopefully I won't, but if I did, okay, if I get a brain lesion, I can't just consider, well, what were the functions now removed? I've got to consider the areas that area was connected to and how much deafferentation that's caused. I will lose midbrain function too if it projects heavily to the midbrain. That's been studied, corticotectal diastasis. All right. What part of the forebrain is most involved in the changes that occur during habit formation? We call it procedural learning or implicit learning. Anybody? It's the corpus striatum. And we'll be talking about that. We already mentioned it. Remember I said early inputs, very early, when olfaction was the main sense of before there were anything except olfaction coming into the forebrain, or the end brain, it was all olfactory. But they went to the striatum. That was the link to the lower structures that control movement. And those connections were plastic. They could be changed depending on the results of the movement. All right, so what about the tween brain? If you look at the just think of the main inputs to the tween brain. And it's relevant to this question. According to the suggestions in the chapter, what is a reason why sensory pathways ascending to the forebrain almost always have a connection in the thalamus? Even the olfactory, actually, to get to the neocortex, have to go through the tween brain. But why would that be? Why don't they just go directly? So let's look at the early inputs. Optic inputs were very early. And this is the picture of it. Here I'm showing them coming into the pineal area, the pineal eye, and here they are. Here's a lateral eye. The most primitive connection there goes directly to hypothalamus. What is it doing? It's controlling the activity rhythm that changes with the day-night cycle, the cycle of the sun, in just about all animals. That is a system that's modulating the entire central nervous system. That's what sleep and waking is. It's a cycle of change that affects the whole nervous system. All right? There's many other system-wide modulations too. The pituitary here, another area where secretions affect the whole animal, the whole nervous system. Okay? And there's more than that. Hypothalamus is affecting what goes through the thalamus. It's a modulator. There are connections from hypothalamus to the thalamus. We'll go over that in a little more detail later. And I think it was that modulation of the whole system that made it was important enough that I think it prevented the evolution of more direct connections. There are a few more direct connections that have evolved, but very few. They're almost all the sensory pathways that go to the neocortex stop in the thalamus. Okay, another part in those decerebrate animals that you're removing is what we call the limbic end-brain. Basically, all the parts that are connected to one structure. You know what I'm talking about? The limbic system is characterized by close interconnections with one portion of the upper brainstem. What is it? You can define the limbic system. Those structures at the fringe of our hemispheres. Limbic means fringe. Okay? They're the more primitive parts that were there before any neocortex evolved and expanded. What was the structure? You didn't get that? Sorry? What was it? Not the neocortex, because the limbic system is more primitive than it was there before there was a neocortex. Okay? It's this structure here. Okay? And I'm showing here hypothalamus and how it is interconnected, not only with the olfactory system, but with all these structures of the older parts of the brain, the whole ancient pallium. We call them the dorsal pallium, the medial pallium and dorsal pallium up here. Medial pallium is hippocampus, and then the lateral pallium, which is olfactory cortex, and the ventral pallium, which also gets olfactory input, includes the amygdala area. Those areas are all interconnected with the hypothalamus, and that pretty much defines the limbic system. And there are structures also in the midbrain that now, to point it out, should be part of that because they are also closely interconnected with the hypothalamus and with the limbic system further forward. All right. I want you to read the rest of these. I'll review a little bit of it at the beginning of the next class, but I will mark this. This is where we stopped, and I will post these online. And we'll be able to finish this quickly next time and go on to Class 7.